You'll also see that the extra credit quiz on natural communities is available this week. So you can take that quiz just to show that you did some of the reading and you understand the natural communities. And feel free to do the reading this week if you didn't get to do it last week. Natural communities is a really wonderful concept for trying to understand the natural world. It helps us to see differences from one community to another and understand the differences, the reasons for the differences. And the natural community concept will be really helpful at your home base this week. So this week the home base is about biodiversity and communities. And when you're out there, it's important that you're actually counting things this week. So you want to count how many different species of trees there are. How many different kinds of goldenrod do you see? How many different birds are you seeing? And then you can move beyond that, of course, and look at community. So that's where the natural community concept is helpful. Do you see a bankside stream community and then also a field community? That's two different kinds of natural communities. You might look at the edge of where those two communities meet and see what you notice there. So this week, pay attention to biodiversity, the different kinds of variety of life, and the natural communities that are there. Your other major task this week is to work on your lab report. So you should have collected the tree biodiversity data in the forest of your choosing and submitted your graphs, and I'll give you feedback on the graphs within the next couple of days if you haven't gotten it already. And now you need to put the work that you did into a full lab report. This lab report doesn't have to be long and involved and complicated. Just try to keep it really simple. Stick to the facts. Just state as plainly as possible what you did. The lab report is organized into sections. There's an introduction section, a methods, a results, and a discussion. And these 4 words need to be in your report as headings for each section. It's usually a good idea to start with graphs of your data, which you've already done. But then when you look at the lab report, it's organized into sections. It begins with the introduction. The introduction sets the scene. It gives the background of your study. So one of the things you do first is explain the major concepts in the study. In this case, it's biodiversity. So you want to explain what is biodiversity, how do you measure it, which means that you're going to talk about evenness and richness and what those things are and how they relate to this particular study. Then you want to talk about the purpose of your study. Why are you doing this at all? In our case, it's either to compare the biodiversity of 2 different forests or it's to compare 2 different sampling techniques. So write about which one you chose and why you chose it. So what is your purpose and why is that important to you? In the introduction, we also include a hypothesis. So you may have had a hypothesis. You may think, well, I think the line transect method is going to do a better job of capturing biodiversity than the large quadrat method, and then explain why you think that is and what you think your results will show. Then move on to the methods. The methods should very clearly and precisely state the procedure that you followed. Just put it in first person and past tense. I measured these trees. I created a transect line that was 40 feet long. Be precise in the information that you give so that someone in this class could exactly replicate your procedure using only the information you give in the methods. So in the methods section, you explain the procedure that you followed. And for the most part, you're going to kind of paraphrase what was in the lab manual. But I know that many of you modified the procedure to fit your particular situation or to fit your particular ideas about how the study should go. And that's perfectly fine. Just explain exactly what you did. Tell us how big were the squares? How many feet of transect did you use? How did you decide where to put your transect lines or where to put your quadrats? This should be written in a way that someone could repeat the procedure, someone who's a peer of yours, someone else in the class, could repeat the procedure just by reading the material that you wrote. The last thing I'd like to see in the methods is a description of the forests. So describe the tree sizes, maybe the ground litter, the location of the forest, and the landscape. So just give a little bit of a forest description so that we have a visual of what this place was like. The next part is the results section. And you've already done the graphs. So that's a great start. So the graphs go in the results, but the other piece that goes in the results is text that explains the graphs. So this text needs to give the richness of both areas, and it also needs to describe the evenness or compare the evenness of the two areas. So it might just say this one was more even than that one, and I know because there are more bars that are the same height in this first forest. Then in the discussion section, this is where you get to explain the reasoning for the results and what those results might mean. So you can say why you think the transect method did a better job of capturing the biodiversity than the large quadrat method. Explain why. And then also you can modify your procedure for if this were to be done again. You'd say, well, since the transect method captured more biodiversity than the large quadrat, I probably need to make the large quadrat bigger in order to have these two have the same result. So give a modification to your procedure to make it even better for the next time. The other thing to do is to think about why is this information even useful? So what is its application? What is its value? Why would somebody want to do this kind of a study? And then finally end with some further questions that you have. What other questions do you have about the biodiversity in this place? What else would you like to find out? So the questions are extremely important to show your curiosity and your understanding of how biodiversity works. So when you have your lab report completed or partially completed, please post it so the rest of us can read it. It will help the rest of the class to have examples of what lab reports can look like. And you'll get extra credit for posting a full or partial version of your lab report. And then students in the class can peer review your lab report. So download the peer review rubric and then give feedback to your classmates. If you post it by Friday, I will also give you some feedback about your lab report so that you can have the best possible product. So take all of our feedback, make revisions, and then post it by Tuesday, October 9. I also added a document that shares how to calculate a Simpson's index, which is a numerical value for biodiversity. So that might be interesting to some of you. You can calculate the Simpson's index for the 2 forests that you sampled, or for the 2 sampling methods, and see how that number compares. So download the document and follow the directions there, and I can help you individually if you'd like that. Please call me. It'll be the